Yeah, thanks for joining us. Um, we're really excited, as I mentioned, to hear what you have um, to share with us because, as I mentioned, it's right at the heart of diversity, and we've been looking at so many systems of growing many human lines and being able to understand and control, and even, as you speak about in your own work, use the diversity of human pluripotent stem cell lines to break apart these early mechanisms in regionalization. Without further ado, take it away. Let us know what you're looking at. Very excited to hear.
Well, thank you for the opportunity to share my work with you, and I'll just briefly introduce myself again. So my name is Magdalena Sutcliffe. I'm a senior scientist in Madeleine Lancaster lab at the MRC Laboratory of Molecular Biology in Cambridge, United Kingdom. And I just initially I wanted to introduce what we do in the Lancaster lab. So as in this slide, the organoids are in the core of what we do, but we sort of incorporate several aspects of diversity and development into our work. And on the left side, these are our experimental models that we then either make or aim to make organoids from, to start with humans, primates, and also other model animals such as mouse, which is great for benchmarking our tissues against in vivo as well. And also we would like to try to extend our work into more sort of the non-model species to open avenues of being able to model brain development in those species. And so here I'm showing a horse because there are ES cells available from horse, but we're also interested in more exotic animals such as, for example, dolphins. And really what interests us is then what determines the size of the brain, and especially the expansion that led to the complex and big human brain. So we look at brain development, at the early mechanism of expansion, at what guides neuronal maturation and outgrowth, evolutionary mechanisms. Of course, then we'll always touch upon disease mechanisms and where my project fits in the method development. So there are always improvements underway. So our lab works on the unguided protocol, and this is our choice because it allows us to investigate particular aspects of brain development that are not possible using other methods. So this method relies on very gently just supporting pluripotent stem cells to exit pluripotency and then fall into the default trajectory of development, which seems to be dorsal forebrain. And as an example, I can give you this publication from a few years back from our lab when we studied the transition between neuroepithelia towards neural glia. So this transition sort of in the organoids happens around day 7 to day 15. And then we noticed that because we don't shorten this time window with inhibition of SMADs, which is the usual approach for neural organoids, we are able to observe this natural pluripotency exit in a slow transition as it unfolds in the specific to species timing. And we notice that it's longer in humans and shorter in our close relative gorilla. And then this slowing down of the transition whilst maintaining quick proliferation rate sets up the progenitor pool for development of larger brain eventually. We also, this is work from Barbara Treutlein, that a lab that compared different organoids, either made with unguided or a guided method. And it just pointed that the unguided method has got more cellular variability, more diversity in cell types, which also allows certain self-regulatory mechanisms to happen within the organoid, whereas as expected, the guided organoids are more targeted to asking sort of specific questions targeted to a specific brain regions. We also put a lot of emphasis on the correct architecture and morphology of our organoids, because in our work, also from a couple of years back, we noticed that the more complex and the more similar to in vivo the architecture and the appearance of the tissue is, the more the transcriptomic signatures resemble the in vivo situation in the brain. So we also put a lot of emphasis on that. And this is how my project and our work links into the developmental diversity in a dish, which is the subject of this seminar series. Yeah, what I'm going to talk today is about the variability, which seems to be the roadblock for many experiments that would like to incorporate diversity in the setup. So this is an example of our perfect organoid made out of our favorite cell line H9, which just performs consistently very well. But there are also examples of cell lines that perform a bit less fantastically, and also of the cell lines that perform consistently bad. And really, the first question at the beginning of my project was, is this something that we just have to put up with? Do we have to select cell lines that are suitable for our model? Or is there something we can do about it? And this is definitely not a new problem, and not a problem that is limited to brain organoid field. Why cells in the embryo are able to, also the epiblast in the embryo is able to produce the entire embryo with no problem. Whereas if we make pluripotent stem cells or derive pluripotent stem cells from embryos, they have a preference to making a particular tissue. And as I said, this is not a new problem. There have been comprehensive publications about this years back, over a decade back, showing biases in both induced pluripotent stem cells and ES stem cells towards producing certain lineages, and then also sort of pointing out towards other problems such as inefficiency, contamination of different identities, completely wrong identities, or cells not surviving their differentiations. So there was this great publication also from another lab in Cambridge here, who looked at differentiation of induced pluripotent stem cells into neurons, into D. So they had a lot of data on what transcriptomic signatures are predictive of neural differentiation success. And also we collaborated with that group, was from the Sanger Institute, and made organoids from their pooled iPS cells. And this was a very interesting experiment because we started with a pool of iPS cells that were grown together even before making an organoid. Then they were dissociated and organoids were made out of them, and then they were matured to over 100 days, dissociated, and then we performed single-cell RNA-seq. And even in this highly controlled environment, where cells were cultured together, underwent exactly the same treatment, and they were even aggregated in the same tissue, they produced their own identities. So here you can see different genotypes, so we can trace the donors. So these are iPS cell lines made from different human individuals. And here you can see the identities that were identified from the cells, and you can see that they really segregate quite neatly. So the cell lines that are in this cloud kind of correspond to those brain identities, and the cell lines that are in this cloud, represented in this cloud of the cluster, made non-brain identities. So we thought, okay, can we then identify those cells' intrinsic properties by the transcriptome of those cells to see what is really wrong with them or what's different about them, and determine whether there is something that we can tell about those cell lines even before they start differentiating, to inform us whether they'll be any good for brain organoids. So we did exactly that. So those cell lines came from a HIPSCI initiative, which is a UK-based bank of induced pluripotent stem cells, and they are fully characterized, all pluripotent, derived under controlled conditions, and also come with a lot of data sets that characterize them for example, bulk RNA-seq. So we were able to link this phenotype of how well they differentiated or not to the transcriptome at the pluripotent stem cell level, and then see what was different about it. And this is kind of very similar experiment to what was done in the study of differentiation of TG neurons, but we really focused on those few cell lines that we can then validate as single preps as well. So when we just did a simple principal component analysis, we noticed that there is quite a strict separation between the competent lines, which kind of clustered here on the left, and the non-competent lines, which clustered on the right. And there were some in-betweeners, so that's why we decided to focus on the most sort of extreme phenotypes and then compare what was different about them. So these are the results of that. This is just the DE-seq experiment, and then we selected several hits and then put them through a GO-term enrichment analysis. And then we discovered a lot of terms associated with broadlead transcription, and then also terms associated with sort of body plan development, things like neural crest or anterior-posterior regionalization. So that was already a hint that there is something going on in terms of starting development, or maybe already expressing genes that should not yet be expressed at the pluripotent stem cell level. And this is something that maybe is happening prematurely. So we had this hypothesis that these cell lines have the sort of posteriorized phenotype. And then to have a benchmark to test that, we turned to stem cell embryo model from, we used the data from Jacob Hanna lab. They made those structures that at day 8 resemble Carnegie stage 5C26 human embryo. So this is the time sort of just at the verge of gastrulation. So we thought that would be the best comparison for our early regionalization, because this is when the cells in the epiblast would be subjected to those regionalization cues. So here's the analysis of this single cell data set. And here encircled in red, you can see the epiblast cluster. And it's sort of regionalized from anterior to posterior. And then cluster 22, the posterior most epiblast, is sort of the beginning of the primitive streak. So then we pseudobulk those clusters and then compare them to our bulk RNA-seq data from our cell lines, which we then early classified as good or bad differentiators. So here in those diamond shapes, you have the clusters of the epiblast of the stem cell embryo model. And if we look from left to right alongside the principal component 1, this is the sort of anterior-posterior gradient captured here. And this is sort of echoed in those, in the top variable genes of our competent and non-competent lines. So the competent lines are the ones that differentiate into brain organoids with no problem, and the non-competent, they produce other tissues. So yeah, this is sort of echoed, and then so here in the sort of the more anterior part, we have the good differentiators, the competent lines, and then the posterior part, we have the non-competent lines. So those are definitely some similarities between this anterior-posterior signature, and we wanted to explore it further. We wanted to see what was the basis of the differences in the transcription. So we wanted to look at what is happening with the chromatin, because that usually determines on what is transcribed, and we just reached out for the first sort of most rudimentary assay, which is a tag-seq. So we just wanted to see what chromatin was more accessible, and then see whether there are differences in chromatin accessibility. So again, when we did principal component analysis on these data, and this is bulk tag-seq, we noticed that the lines that were competent sort of clustered together, and the lines that were non-competent clustered separately. So that was quite promising, that there are differences in chromatin accessibility. And then we looked at the peaks that were more accessible and checked for enriched motifs in those peaks. We saw that they were mainly in sort of regulatory regions, mainly in introns and intergenic regions, with only a minor fraction in promoters. And then when we looked at the motifs enriched in those peaks, we noticed that especially in the non-competent lines, the enrichment was mainly for homeodomain and forkhead, and also SOX factors, which all ring a bell as developmental transcription factors. So that was kind of in line with the DSeq analysis that we did before. So that suggested that there is some sort of opening of the chromatin, something going on in terms of development. So we thought that if there is a difference at the chromatin level, then this is at the epigenetic level, that this is potentially reversible, and we could find a method maybe to restore the chromatin structure to this competent state and make those cells have a chromatin landscape more like competent cells in the non-competent lines. So this is where the method comes in of restoration of competent like chromatin. So the method is basically a three-step method of a cocktail of small molecules that in medium 1 target a range of chromatin modifiers. So it ranges, basically disrupts the chromatin structure by inhibiting a lot of histone modifiers, and histone deacetylases, also targets DNA methylation directly by MEK inhibitor. And then the remaining 2 media are the sort of recovery media where cells are maintained in this kind of almost like naive-like conditions and then slowly transition towards prime conditions with addition of FGF2 and Albumax and withdrawal of MEK inhibitor. And then finally, the cells are maintained in the usual medium, which for this we use Stemflex on laminin 5:1. So actually the extracellular matrix here is also important because laminin 5:1 has been reported to maintain the best sort of pluripotency factor expression profile. And then we maintain IWP2, which is a Wnt inhibitor, because this comes from this idea of anterior to posterior gradient in those cells, and Wnt is the main factor that actually leads to this process in vivo and also in vitro. So
Magda, could I ask you a question quick there?
Certainly.
So that is quite a complex multi-step cocktail. How did you arrive at this?
So this is
you can abbreviate the process, but was there screening?
Yes,
Was there
yeah.
guessing?
So that was a modification of a part of a method that was used to chemically reprogram somatic human cells to induce pluripotent stem cells. So I had this idea that if this can, so in that method, I can't remember. I can't remember the name of the group. It's cited in my manuscript. They have sort of intermediate steps and of cells sort of ranging from somatic cells that they treat with different cocktails of inhibitors. And basically at the final stage, they have something that's almost pluripotent, but not yet. So this is the sort of this step with certain modifications and additions that I took from literature and sort of experience in the lab that kind of just takes our pluripotent stem cells and just, sort of probably erases something in the chromatin and then puts them back in those perfect conditions with laminin 5:1 Stemflex with inhibition of Wnt signaling, so they don't really see those posteriorizing signals that they can secrete themselves. And that sort of stabilizes them in this more anterior phenotype that we think is compatible with brain organoid differentiation.
Thanks.
So yeah, definitely built upon prior art.
So can I ask one more question related to that?
Sure.
So in your cocktail, you have IWP2, which is, as you said, it's a Wnt inhibitor, but you also have the chiron molecule, the Wnt activator, right? So how
Yes, well spotted. So the concentration of chiron here is 0.5 micromolar, and the concentration that is used to actually inhibit or promote Wnt signaling is usually in the range of 3 micromolar plus. So this is a GSK3 beta inhibitor, right? So it probably captures other functions of this protein in maintaining pluripotency other than Wnt inhibition, sorry, the Wnt promotion. So,
okay.
uh,
Thank you.
okay, let me just, okay. So, this was sort of the first test that I put my restored cells to. So that was kind of the readout whether the method even worked. So let me just guide you through this figure. So these are examples of brain organoids or kind of products of brain organoid protocol of those lines that didn't want to make brain organoids in the first place. So these are tissues that should normally look like the right panel, but they just haven't got the right marker expression on the right structures. They should have those SOX2 positive rosettes. They should have TBR2, which is a marker of dorsal forebrain, and also by DAPI, the general morphology of sort of neural buds. And they definitely haven't got any of that at day 20. So at this stage, those tissues are formed and they're never going to recover, they're never going to get better. But after I subjected those cell lines to the treatment that I described in the previous slide and then made brain organoids out of them, they actually behaved like our competent lines. So they produced neural buds that are SOX2 positive. They do not express SOX10, which is a marker of neural crest here, or ecadherin, which is a marker of surface ectoderm at this stage. And then on day 20, they start expressing TBR2, which is the marker of dorsal forebrain. So they are producing, sort of acquiring dorsal forebrain identity. And then they sort of follow this development and expand the ventricle-like structures, again with nicely lined with those intermediate progenitors. So these, if I didn't know which lines they were made from, I would say that they're probably made from our competent H9 cells because they behaved really well. So that was definitely success that I managed to restore this organoid competency in those lines by putting them through this treatment. And so the next question was, are those cells now biased towards anterior or are those cells just sort of free of any biases? So this is when we tested them for sort of general pluripotency marker expression, but also what is important here is that it wasn't the pluripotency marker expression or the growth of the cells, but it was their differentiation potential that was suffering. So I really wanted to test that. So I tested them in assays where I differentiated them to mesoderm derivative kidney organoids and then endoderm derivatives intestinal organoids. And here in the left panel, you have the cell lines before treatment, and then in the right panels after treatment. So they behave way better in kidney organoid differentiation and also in intestinal organoid differentiation. So the difference is striking in brain and kidney, a bit less pronounced in intestinal organoids, but the tissues generally have better marker expression and colocalization of CDX2 and ecadherin, which is indicative of intestinal tissues. And then we really wanted to characterize the mechanism of what is really happening with this method. So when we then again looked at a TAC-seq of those restored cell lines and compared them to the parental lines. So here you'll see that we have matched parental circles and then treated cells are triangles. So the same color is the same cell line before and after the treatment. And here I included stem cell line HECT1, which is a naturally competent line. So this iPSC line produces beautiful brain organoids. I included it in my treatment to see whether application of the treatment on already competent line is detrimental, or is it neutral, or does it improve the performance of the cells? And I can tell you that it is actually neutral. So it basically, from a competent line, it produces a competent line again, and from non-competent line, it produces a competent line. So the effect seems to be similar every time. But in this plot, you can see that when we have this competent line sort of clusters towards the left side of the plot after and before treatment, which sort of should reflect similar chromatin accessibility profile in both before and after treatment. Whereas when we have non-competent lines, such as the red one, the yellow one, or the blue one, they cluster closer together before treatment and then closer with the naturally competent and restored lines after the treatment. This is also visible when we look at the transcriptome. So again, here's the same story, that we have lines after and before the treatment. So the parental lines are circles, and then the treated lines are triangles. And then we also have our favorite H9 cell line as a positive control of a competent line. You can see that the transcriptomes of all those restored and also naturally competent lines cluster closer together than the transcriptomes of the non-competent lines. And then we went back to the stem cell embryo model to see whether the genes that went down after the treatment in the non-competent lines, whether they had any sort of expression signature in this data set. And we noticed that they were overrepresented here in those posterior clusters of the stem cell embryo epiblast. So these are the genes that were downregulated with the treatment, which sort of fits in the idea that those cells have aberrant expression or premature expression of posterior genes. And really to dig deep into the mechanism of what is happening, to kind of know that the chromatin accessibility changes, the transcription changes, but we couldn't quite explain why the transcription changes, because the ataxic peaks were mainly present in the regulatory region, so they seem to be in sort of introns and also in intergenic regions. So we wanted to see whether we can find a direct explanation of the transcription differences. So here we look at bivalency. So I guess everybody is familiar with bivalency and the epigenetic marks that determine bivalency, but I'll just make sure that we're all on the same page. So histone 3, lysine 27 trimethylation is usually enriched on genes that are not expressed, and then histone 3, lysine 4 trimethylation is enriched on genes that are actively expressed. So when we compared the methylation on the subset of genes that changed the most after treatment and looked at those inhibitory and active marks in our cells and their parental counterparts, we saw that those naturally competent lines and cells after treatment had enrichment of inhibitory mark on those genes that are downregulated, whereas the parental lines had lower signal in those genes. The difference was also partially or slightly visible in the level of the active mark. Apologies. In the active mark, but not as pronounced as with the inhibitory mark. So we can see higher level of histone 3 lysine 4 trimethylation in the non-competent lines on those genes that went down after treatment, and then slightly lower levels in competent lines and then in restored lines. So that suggests that there is regulation at the level of bivalency on those developmental genes that determine the competency. So if we zoom in on a particular locus, which is differentially expressed, differentially modified, and not differentially accessible, I can talk you through the differences at the epigenetic level. So here you can see that there is RNA signal in the non-competent line before restoration, and it disappears after restoration. There is little correlation with ATAC signal directly in the locus. So it's difficult to explain the differences in expression with just chromatin accessibility. But if we look deeper at the histone modifications, we see that the inhibitory mark is missing in the non-competent line, according, sort of in line with presence of the transcript, and then it's restored after the treatment, whereas the active mark is higher before treatment and goes down sort of to maintain bivalency level and maintain the locus poised for activation after the treatment. So this is the sort of state that the developmental genes should be in pluripotent stem cells. And then finally, it's sort of my penultimate slide that takes me to sort of conceptual model of what is happening here. So we think that competent pluripotent stem cells are more similar in their epigenetic state to anterior epiblast, which is capable of producing both brain and also posterior epiblast through gastrulation. But the non-competent pluripotent stem cells might be on their way towards this posterior epiblast phenotype, which is under natural circumstances irreversible, but with application of the chromatin restoration treatment, this more posterior phenotype can be then reversed to more anterior phenotype, compatible with all possible epiblast differentiation. And there is also a gradient, a gradient from anterior to posterior in developmental gene expression, developmental enhancer opening, bivalency, and differentiation potential. So before I take questions and we open the discussion, I would like to acknowledge people involved in this work, especially Stephen Wingett, which is our bioinformatician and has done a lot of work on analyzing all the omics data that I have here, and also Eugenia Wong and Stefan Schoenfelder from the Abraham Institute near Cambridge, and they're really the experts on bivalency and histone modifications. So thank you very much for your attention. I'll be very happy to discuss and take your suggestions about this.
Thank you, Magdalena. And please jump in with questions.
I'll just keep the screen share for now in case we need to go back to some slides.
Yes, I think it's a good idea. Go for it, Joshua.
Hi, Magdalena. Really amazing talk. A question would be about how stable is this reset state, basically? Once you revert the epigenetic state, is that something that you can keep in culture, or would you do this for each experiment as the starting point?
Yes, so fortunately, it is possible to maintain it for the longest I've tested it so far is 15 passages. So I kind of accounted to IWP2.
Did you say 16? Sorry,
15, 1 5.
Thanks.
So yeah, I attribute it to sort of presence of IWP2 because if really it's the Wnt that is the culprit here, and we extinguish the signaling that should keep those cells in the right state.
So through those 15 passages, you kept the Wnt inhibition around?
Yes.
Okay.
Yeah.
Without it, does it revert?
So I haven't fully tested that yet, but
Okay.
yeah, I kind of expect that it would. Yeah, I probably have to do those experiments. I'm sure that the reviewers are going to ask for that eventually.
Jason, go for it.
I wonder if you tried sort of just using a guided differentiation instead of the unguided differentiation, and if that would lead to sort of more consistency across different pluripotent lines. Because it seems to me like, I don't know, that there was too many chemicals that I didn't see them. I didn't pay attention to all of them. But like, like a TGF beta inhibitor and a Wnt inhibitor are commonly used for guided differentiation protocols, and at least you have the Wnt inhibitor. So are you kind of doing a guided, like sort of setting them in a guided state?
Yes, so we are very focused on the unguided differentiation. So, but I haven't done those experiments myself, but those cell lines also failed in guided differentiation. And also Eugenia and Stefan have a project where they try to differentiate those cell lines into neural progenitor cells and then see whether those epigenetic marks persist. And they're really having difficulties to differentiate those non-competent lines. So that kind of suggests that it's kind of universal to neural in general. And also, there are people who struggle with guided differentiation and consistent performance. And I think probably it will be fewer lines. It's probably less sensitive if it's more guided and provides more cues on how the cells should behave. But I don't think it's completely free of biases, because other protocols for other germ cell derivatives also suffer from that, and they can be produced through unguided. And I think you suggested that I could try to do guided differentiation with Wnt inhibition. And I think people do also try that. And I'm not sure whether it's a silver bullet either. But yeah, I definitely agree that guided differentiation is probably helpful in some circumstances.
So are there other pathways? Like, so I didn't see like all the, which particular pathways are you guys inhibiting? Like, so I know that was the Wnt and
Yeah, so I think what I kind of really want to emphasize here is that we don't inhibit those pathways with generating the organoids. We inhibit those pathways in the transient state when we transition the non-competent lines to competent lines. And then we sort of just culture them under normal pluripotent primed conditions with Wnt inhibition, but then they go into unguided protocol again. So it kind of just erase the starting material, and then they are then suitable for unguided. So those molecules are not used in the organoids protocol. They kind of used as to say, like reprogramming step.
Just in the pluripotent. Okay, gotcha. Thank you.
Hi, can I go next?
Go for it.
Hi, so I'm very interested in your chip sequencing data with H3K27 trimethylation and H3K4 trimethylation.
Mm-hmm.
And there, maybe I missed this, but when you're showing the accumulative peaks where
Yes.
Yeah. So here, are we looking at all genes that have the K27 23 peaks?
No, so I think I omitted that from the explanation. So these are cat and tag experiments. They're not ChIP-seq, but I guess the outcome is kind of similar enough.
Yep.
So these are the genes that specifically go down after the treatment. So it's not all the genes. I haven't got the profile for all genes in this presentation, and it seems to also differ slightly, but not as dramatically as in the genes that actually change expression after the treatment. And I also have a profile of all genes, but the ones that change, and then you can hardly see any difference. So it doesn't seem to be a global change. It seems to be a sensitivity in particular gene set, which we're kind of also trying to identify to characterize which genes are under this pressure of losing bivalency.
Got it. And do you know which small molecules that you're using could be specifically targeting K27 trimethylation or K4?
Yes. So in a way, the cocktail that I'm using is not directly targeting this.
Right.
So that's why I think that the process is more sort of completely erasing the chromatin, or completely sort of making this sort of plastic chromatin, which you then stabilize, rather than targeting particular modifications that we know that we then sort of retrospectively learned were different. So this is something, those profiles we learned after we tested the method and we knew it worked. So it wasn't sort of informed by this bivalency loss. But I guess it's interesting to see whether if we targeted some of those modifications directly, we could maybe achieve better results even.
Okay, thank you.
And Pao?
Okay, I have two quick questions. Thanks for presenting. I think it's amazing because we also work with Hipsy lines and we have the same problem most of the times. And we work in 2D, not in 3D. And I was wondering, when you made the organoids, don't you have a problem where the non-competent lines force somehow the competent lines into go into the non-competent line direction in the differentiation? Because that's something we see quite often in our cell villages.
Yes, so I'll just go back to those data here. And
Because you are able to produce neurons actually, right? In this UMAP, you have neurons of the lines that are competent.
yes, so actually, what we saw here is that the split was really, the identity was really cell line specific, but I can imagine that those non-competent lines, because some of them even, I think they were like Wnt secreting phenotypes, so I'm sure that some of them could potentially lead the good ones astray by this rogue signaling in the setting. But I haven't probably got enough evidence to say that. We have some cell lines that are kind of the in-between phenotype here, and then it could be that if we did a single cell line differentiation, they would behave better, but we haven't fully investigated that to sort of confirm that.
Okay, well, we can talk about it because that's we are doing
Okay.
experiments. We might see whether we are on the
Is
same.
is that in brain organoids specifically
No,
that
no, no,
you?
we are doing it today. We don't have 3D expertise.
Okay. Oh, but this is also in neural.
Yeah, yeah.
Okay.
We
Okay.
use the legacy protocol, the Seattle one, and we have these issues. And the second quick question is that I was wondering what's worth here, because I think it was Joshua or Jason that asked about the stability of the
Mm-hmm.
lines after restoration, but I wonder about the stability before restoration. So about the same IPS lines we use of these big resources. Do you think they lose this stability at some point and they have regionalized with the time with passaging or not? Because then my question is worth trying to restore the chromatin or generating new IPS lines of the same donor? That's like a key question, no? I mean, what's where we have to put more effort?
Yeah, so I think there are two mechanisms at play here because, and also, kind of two aspects of this question. So maybe I'll start with the sort of quicker one. There are some cell lines where you haven't got the donor cells anymore. It's some sort of precious patient line, and you want to just make them work for you. So I guess this is for this sort of situation. I guess if you can go back to the starting material, then it might be maybe easier, maybe more efficient to reprogram them again. And I guess this work provides you with some information of how to prevent the sort of relapse. But this seems to also happen with ES lines. So here, for example, we have US8 line, which also didn't make organoids in our hands. And now we also have the RNA-seq and cut and tag data from these. And they have the very similar signature to those non-competent IPS line, which was particularly important for us because it gives us a sort of more general mechanism. Because in IPS, you could argue, oh, this is just incomplete reprogramming, and then we should just try a different clone. Because interestingly, I haven't tested that myself, but that was in Julie Gerber paper that we collaborated on with those organoids, that there were lines from the same donor, different IPS lines that had predicted different competencies. So one of the lines was competent and the other one was not. And so that could be because one was reprogrammed better, but also because um, especially the K27 trimethylation is a target of PRC2, and I think I found two publications from about a decade ago, about 5 years ago, that mentioned that PRC2 targets are the most variably expressed genes in between iPS cell lines. And I think one of those studies was done on identical twins and cells reprogrammed from identical twins, and then they saw that the most variably expressed genes between kind of almost genetically identical individuals had varied levels of those genes. So there's clearly some pressure on the PRC2 targets, and
Could you share that reference with us when you dig it up?
Oh,
I'd love
yes,
to bump
certainly,
into that.
yes.
Thank you.
Is that on Slack that you share references?
Sure, you could do it there. That'd be great if you're connected.
Okay, I'll have to join. I haven't done it yet. So yeah, so I think there are two papers. One is cited in my manuscript, and the other one actually I found probably about a month ago. So that was also reassuring that there is sort of the mechanism is kind of already reported.
Very cool.
Thanks so much.
Ron,
Thank
jump
you.
on in here.
Hi, great talk. And tons of interesting points being discussed here. But this, if we can just focus on this slide that you have up at the moment.
Oh, perfect. Yeah.
So I just have a few comments and there are questions interspersed in here. So first of all, is it correct to think of this as suggesting that the biggest effect of your resetting these cells is actually on the H3K27 trimethylation signature?
Yes.
That's really what's different. Yeah,
Yes.
okay. Yeah, great. And then maybe I could ask you a question or two about what genes might be involved. So do you see Hox genes showing this change?
So no. So I think
great. That's great.
So I think the only Hox gene that shows that, and I don't want to be, so it's very interesting because the genes that show this difference is actually GBX2. So if you look at if you look at the homeobox gradient, if you were looking from anterior to posterior, we'll have OTX2, GBX2, and then the Hox cluster, right? And then I think that the first Hox gene, if I'm not mistaken, is it Hox B2, which is the anteriormost hindbrain. So this is
yes, that's a great response. I mean, I think the details matter, but overall, it isn't. You're not reading out a kind of a Hox linear code straight off. It's slightly more complicated than that. But GBX2,
slightly more anterior.
I mean,
yeah. Right.
Well, I mean, the question, I think, really, I mean, to some extent, our observations here may be not quite as systematic as yours, but we came upon GBX2 as being a key determinant of this transition that you're looking at, which you call competent incompetent.
Okay.
But I wonder if it's not Hox genes. Have you also looked at NMP genes, the neuromesodermal progenitor genes, precisely in this data set? I mean, maybe we could come back and talk about this later on, but I think it's extremely interesting what this is.
So actually, I was, because I kind of got lost into trying to remember which Hox gene that was, but it's the anteriormost. If you look at the gradients of Hox expression, it's the anteriormost Hox gene that already has evidence of loss of K27 trimethylation, but yet is not yet expressed. But GBX2 is expressed. So it seems like the gradient is a sort of opening expression, and then at some point we don't have expression because we don't have enough K4 trimethylation, but we already have evidence of sort of dampening of the inhibitory signal. So that would show that there is this sort of posteriorization happening, but it's not very advanced. And if you think about the sort of anterior signature, it really is the difference between OTX2 and GBX2. That is the sort of first border that forms, at least, I mean, in mouse embryos, that's what is better investigated. And then you have the activation of Hox sort of posterior. So it's not yet Hox, it's the anterior class, it's sort of more anterior homeobox genes that are under pressure here.
Well, it's the midbrain hindbrain border in mouse, right? This is Alex Joyner, and
Yes.
but it's also, I think, of great interest to ask again about the NMPs, because in the I don't know if you're probably as familiar with this as many of us are, but in that Pijuan Sala data set about mouse gastrulation, it's very clear that the NMP population is appearing very early. It's almost like right at primitive street kind of time. So maybe we could discuss this and even look at it together. That would be a very interesting thing to explore in these data.
Yes.
Anyway, thanks again. Wonderful talk. Great.
Thank you very much. Very good questions. Thank you.
Could I ask one more quick one?
Jump on in.
Did you find it that the qPCR scorecards were predictive at all? The qPCR scorecards of the pluripotency measures of gene expression were predictive, especially like those genes that are measured in the qPCR scorecard, were predictive of the differentiation potential of the iPSCs.
So that's a good question. I haven't actually compared them, but I think it would be interesting to look at that. So because I guess it's because here I only have, I'm not showing this. It's kind of an image to show that this is something that was recognized way back. I think it's a good idea. Also, so we're kind of almost now arriving at the sort of layers of genes that tend to be aberrantly expressed and that correlate with how bad a cell line is. So it seems to be a bit of a gradient, and it seems like that some of the cell lines express sort of even more and more and more posteriorized genes, and some of the cell lines only express this kind of slightly posteriorized genes. And what is really interesting, in some of those cell lines, actually one in particular, I was able to just get rid of that signature just by inhibiting Wnt. So probably if there is like not enough Wnt signal just being thrown at those cells and you just inhibit that, they kind of with the cell divisions, that signature will get diluted. And then if it's really sort of ingrained and it's gone far and far away and it led to expression of a lot of aberrant genes, then it really needs some sort of harsh action with those small molecules. I guess it would be interesting to compare those data sets that we kind of arriving at and the scorecard genes. And also there were some hits from Julie Gerber paper, but because I think there were a lot of cell lines and probably there was just almost too many hits to sort of come to some conclusions, but there are some overlaps. For example, UTF1/2 seems to be a general predictor of poor differentiation.
Thanks.
Can I just keep the conversation flowing a little bit here, especially as your kidney result I find very interesting. That when you sort of reset the cells, they now appear to differentiate towards kidney more efficiently. Is that correct? Because that at first glance doesn't appear to fall into an anterior posterior kind of mindset. But I'm wondering whether it's more correct to think of this as being a choice between a sort of forebrain and gastroid kind of fate. And that is that a good way of thinking about it?
Yes, I think it's a very good way of thinking about it, because really I feel so if you sort of try to look at development and how the epiblast sort of portions get directed towards gastrulation, what is left at the end becomes the forebrain or becomes the brain, right? So eventually the part that was never exposed to any signaling then has only got one root left, and it's probably the forebrain. But then going back to the kidney question, I think this is more of a technical aspect of this, because also the effort of this is to sort of standardize, not only to make the cells suitable for cerebral differentiation, but also to standardize the starting material, so that, for example, in cell line that is more sensitive to wind, you would, for example, have to induce a kidney organoid with a lower dose of chiron initially, because maybe they have lower bivalency on the key genes, which will be then activated by chiron. And then you would have to, it's particularly visible in gastruloids when you have to titer initial concentration of the Wnt activator towards the cell line. And this could also come from the same mechanism, right? So if we have sort of the same level of bivalency, high bivalency in those developmental genes, that you could imagine that you would have to use very similar concentration of cheer to induce your sort of intermediate mesoderm to then produce the kidney organoid. And then it is possible that the protocol that was optimized on a competent line for making kidney organoids has just too high cheer. And then if you add that, you produce something that's way past kidney in terms of, the wind landscape, and then it gets different growth factors that would work on the kidney primordium, but it's the wrong environment, so then you don't get the right tissue.
To ask another gastroloid type of question here. So I find it very striking listening to the gastroloid community, especially initially by Alfonso, but where it was clear that there were a certain number of cells where you were optimal for making a gastroloid. And I'm wondering whether you find that's true for your optimization of forebrain fates, that actually it's highly tuned to a certain size of organoid.
Yes, so that's
Would
also
you-
true. Yes, that's also
Right.
true. We've experimented with different sizes of organoids, or sort of different initial cell number for the embryonic body. That is the first stage of the organoid protocol. And we have, so generally the smaller the embryonic body, the better. I think this is also something to do with the fact that maybe the cells inside produce more sort of Wnt or BMP signals, so that would interfere with this process. So the smaller the center is, the better. You can also achieve that by sort of flattening the tissue on microscaffolds, but there's generally sort of top range that you can't go over, and because you won't be able to produce good
How
organoids.
small is small? Is small 300 cells by any chance?
I know in gastroloids that's the
Yeah, right, exactly.
but no, so in we also have then think that you have to have a certain number of cells. I think the smallest that we've gone is like about 200-300 cells, but
Yeah,
I think the
exactly.
optimal is between 2,000 and 9,000 cells.
Oh, okay. Thank you.
Go for it, Paul.
Just one quick question. Have you tried to perform like disease modeling with these CHR restored lines? Because I wonder, you said that you somehow standardize the starting material, but I was wondering if this has an impact also on the disease biology itself, right? Because we want to see differences between donors linked to the disease, but maybe the noisy ones, It's
Yeah, so I guess so then okay, just want to make sure that I
Are we
say it
removing the
correctly.
disease component with this method? That's my question.
Yeah, so I think that is actually should be beneficial for disease modeling, because if you have cell lines that, because of some disease mutations, have, for example, elevated particular pathway that would stop them from differentiating into the tissue of choice, then you can't really model this disease. But because it works on an epigenetic level and then the genetic so if it's it's a disease with a genetic basis, then you can still model that. If it's a disease with epigenetic basis, I'm not sure whether this would be applicable. But in a way, this method allows us to sort of circumvent the problem with the cells developing outside of an embryo context and not having all the sort of safeguarding mechanism, because of course, the cell lines probably acquire some mutations in the process of being cultured and reprogrammed, but they all came from healthy donors that had perfectly developed brains. So, their genetic background should not stop them from developing brain, in theory, in vitro, unless the the system is so very different.
Thanks.
Any more questions? I've really enjoyed this discussion. Well, thanks so much for joining us. As you can tell from the active questions, it's right at the center of what a lot of us are thinking.
Thanks. It's great to talk to people who are in this field and ask really interesting questions and raise very interesting points. I very much enjoyed it, and yeah, thank you so much again for giving me this opportunity. And I love to follow up on those questions, definitely. I think yeah. So I'll join the Slack channel to share the reference that I mentioned. About the PRC2 targets, and then yeah, if anybody would like to follow up on anything or get in touch, please do. You also have my email in the message that Carlo circulated earlier.
Wonderful. Thanks so much.
Thank you.
See you all next time.
Okay. Thanks. Goodbye.
Thanks.